Hi, really nice talk. I'm Rebecca Earnshaw. I'm a PhD student in the Dr. Sunil Kalia lab, and my project is focused on C-terminus of HSP70 interacting protein, or CHIP, in mitochondrial autophagy or mitophagy, and as well in spinocerebellar ataxia. So CHIP is an E3 ubiquitin ligase and an HSP70, HSP90 co-chaperone protein. Its major role in the cell is described as ubiquitinating and targeting proteins for degradation by the proteasome. And interestingly, it's been demonstrated to be able to promote the removal of many disease-associated proteins, including aberrant forms of alpha-synuclein LRRK2, tau, A-beta, SOD1, polyglutamine expanded proteins. So there's a lot of interest in this protein in the neurodegeneration space. And one interesting, mutations in the gene which encodes CHIP is the cause of two forms of spinocerebellar ataxia, which are neurodegenerative diseases resulting in severe cerebellar degeneration, as well as ataxia, which is an impairment in coordination and cognitive impairment. And one thing that is quite interesting is that animal models which have a loss of CHIP display mitochondrial abnormalities, but the reason for this is unknown. So that led us to wonder whether mitochondrial dysfunction may be a component of CHIP-associated neurodegenerative diseases. So like the previous talk, we are also interested in mitochondrial autophagy or mitophagy, which is the selective degradation of mitochondria via macroautophagy. And this occurs normally under basal conditions to just turn over mitochondria, but can also be elevated in response to stress so that the cell can remove damaged mitochondria. And increasingly, dysregulation of mitophagy is being implicated in many neurodegenerative diseases. And so, as described also in the previous talk, one commonly studied mitophagy pathway is the PINK1 Parkin mitophagy pathway, where PINK1 serves to sense damaged mitochondria, which recruits Parkin. Parkin then basically tags the mitochondria for engulfment by the phagosome, which can then fuse with the lysosome. And the lysosome has acidic conditions and hydrolytic enzymes inside of it, which allow it to degrade the mitochondria and remove it from the cell. And so interestingly, CHIP has been demonstrated to have known effects on both PINK1 and Parkin, but no one has really investigated the effects of CHIP on mitophagy yet. So to assess the effects of CHIP on mitophagy, we used a coral-derived pH-dependent fluorescence protein called MT-chyma. And so this protein is normally excited by a short wavelength of light when it's under neutral pH conditions. So when the mitochondria is in the cytosol or in the phagosome, you get short wavelength excitation of this reporter. But when the mitochondria is engulfed in the lysosome, it's exposed to acidic conditions. And it undergoes a conformational shift, causing it to be excited by a longer wavelength of light. And so that is measurable by flow cytometry. And this is just a representation of what the flow cytometry output looks like, is that along the x-axis, you have mitochondria at neutral pH, and along the y-axis, you have mitochondria under acidic conditions. So you can see with the addition of CCCP, which, as described previously, is a potent inducer of mitophagy, you see an upwards shift in the plots, indicating more mitochondria engulfed in the lysosome. And so we use cells expressing this reporter, which were obtained in collaboration with Dr. Fawn at McGill. And cells expressing this reporter were either transfected to overexpress CHIP, or to knock down CHIP and then treated with CCCP, and flow cytometry was used to assess mitophagy. So what we found is that when we overexpressed CHIP, we actually saw a significant decrease in the percentage of cells undergoing mitophagy. And when we knocked CHIP down and treated the cells with CCCP, we saw an increase in the percentage of cells undergoing mitophagy. indicating that CHIP expression is able to alter mitophagy in response to stress in vitro. And so we wanted to take this forward and look to see if these effects were also demonstrated in vivo. And to do this, we used C. elegans. So C. elegans are microscopic nematodes. They're actually quite popular for the assessment of neurodegenerative diseases because they have a short lifespan. So it's very quick for us to assess age-related phenotypes. And they are also very inexpensive and easy to maintain. They're also transparent, so we can directly visualize their neurons while they're still alive without any need for any dissection. And they have an extremely well-characterized nervous system. So this makes them a really good model before moving to a rodent or a different model. And so in this case, I used worms expressing the mitorosella mitophagy reporter protein. So this is a protein that's localized to the mitochondria and it's fluorescent. So it's comprised of a green fluorescent protein or GFP, which is sensitive to pH, and a DS red, which is a red fluorophore, which is pH stable. So basically when the mitochondria is in the lyse or is under neutral pH conditions, like in the cytosol, you get both red and green fluorescence at the mitochondria. But when the mitochondria is engulfed in the lysosome, you get quenching of the green signal. So you only get red fluorescence. So basically, mitophagy is a measure of green to red ratio. And a decrease in this ratio indicates more mitophagy. So we have C. elegans, which express this mitophagy reporter within all of their neurons. And this worm line was generated by Dr. Tavnarakis' lab. And so what I did is I took worms expressing this reporter in their neurons, and I crossed them with worms which have various loss of function deletions in different genes. So what I found was that worms which have loss of function deletions within the CHN1 gene, which is the C. elegans homologue of CHIP, actually displayed a significant decrease in their GFP to DS red pixel intensity ratio, indicating that they have an increase in neuronal mitophagy. And even more interestingly, these last two bars on the end, when I have worms which have this mutation in the CHIP homologue, as well as within the pink homologue, you don't see this increase in neuronal mitophagy, indicating that this is dependent on PINK1, as well as when you have mutations in both CHIP and the Parkin homologue, you again don't see this increase in neuronal mitophagy. So this process does seem to be dependent on both PINK1 and Parkin within worm neurons. So just in basic summary, is that I've found that CHIP expression does alter mitophagy in vitro in cell lines in response to stress, and also appears to alter it in vivo in C. elegans neurons. And so one of the future directions that I'm currently working on is looking at assessing how disease-associated mutations affect mitophagy in vitro, as well as in vivo in C. elegans neurons. So we've generated some worms which have patient-associated disease mutations, which we plan to assess for mitophagy dysfunction, as well as neuronal dysfunction using these worm models. And I just want to thank everyone in the Dr. Sunil Kalia lab and the Dr. Lorraine Kalia lab. I want to really thank MITO2i for their support of my work and for the opportunity to speak to you today. And thank you very much for listening.